All right. Well, our first topic for this term is going to be obviously the muscular system. So you are not responsible for this quote at all by Arnold Schwarzenegger. Just something interesting, I suppose. So I'm going to go through this and hopefully give you an idea of what this system does for us. And the focus is going to be primarily on skeletal muscle, but muscle overall. Basically, it has many functions, right? Most people think about the muscular system, they're thinking about movement and producing movement. Like, for example, the running or walking or turning your head or reaching out to pick something up. All of that requires us to contract skeletal muscles that then, when they contract, they pull, most of them, on their bones to which they are attached. Now, the exception to this would be facial muscles. They're actually attached at one end to the skin, the tissue deep to our skin. But when it contracts, it produces movements of our face, just like the muscles that are attached to our bones. They produce movement. And even if you can, this is skeletal muscle that's producing this type of movement. And actually, if you're sitting right now and watching this, then you're maybe not really moving a whole lot, right? But that's still using your muscular system. Your muscular system is involved in stabilizing your position, being able to stand still, quote unquote, which actually is a misnomer. It's never actually possible to stand 100% still, but that's neither here nor there. Okay, it's the consequence of living on a round planet that's spinning about an axis. We're going to go with science, by the way, and assume that the Earth is round. All right. But we need to contract these muscles all the time to hold a position, to maintain our position in space. All right. So they got movement, you got stabilization, and like I said, there are different types of muscle tissue: skeletal muscle, smooth muscle, cardiac muscle. Right, now, cardiac muscle, it's a special type. It's associated with our heart, and it's involved in, again, contracting, producing movement that actually helps squeeze blood through our heart and effectively send it to our lungs, and then it comes back to our heart, and our heart contracts again and sends it out to our bodies and delivers oxygen and nutrients and picks up waste products and then moves it back throughout our body. And that blood travels through blood vessels. And that's what this is, a cross section right here of a blood vessel. This would normally be filled with blood. And then this layer surrounding it is smooth muscle that squeezes on the blood and helps propel it to where it's supposed to go in your body. This is showing here the stomach. Our gastrointestinal tract is also lined by smooth muscle. Same function, movement, right? You put something in your mouth and you chew it up by contracting your skeletal muscles and crushing it and mixing it with your saliva, and then you swallow it. And this muscular tube of your esophagus is going to move it down into your stomach. Your stomach's going to contract and crush that food up and mix it with all your gastric secretions, and so you can chemically digest that food. Then you'll move it into your small intestine, and that muscular tube will move it through your body and allow you to extract nutrients from it. Okay. So, again, all kinds of different functions here. Now, one of the functions of blood is to deliver nutrients and waste products, and that blood is moved by way of muscle activity. So, one of the major components that's delivered in our blood is glucose. Right? It's like like a blood sugar. You can think of it that way. So in that sense, muscle also plays a role in controlling blood sugar. Right? How it can deliver blood to certain areas. Some of that glucose is actually stored inside of our muscle compartments. So because muscle always contracting takes a lot of energy, we get a lot of energy from breaking down glucose and sugars in order to produce ATP, which is adenosine triphosphate, considered the energy currency of the body. And that's something you should have picked up in Biology 231, but here you got it again. And the last role here is thermoregulatory function performed by the muscular system. If you get cold, right? If you walk outside and you're really cold, you might notice you start shivering. All shivering is, it's rapid contraction and relaxation of skeletal muscles. And I'd like to say this isn't the case, but we're actually not that efficient at using all of the energy produced by our skeletal muscles. A lot of it is, I wouldn't say wasted, but it is released in the form of heat, right? So if you get a lot of muscle contracting quickly, it's going to produce a lot of heat, which if you're cold, that extra heat, shivering, will warm you up. It will raise your internal body temperature. Right, so these are just some general functions of the muscular system in general. There you go. I did mention there are these different types of muscle tissue, 3 different types: skeletal, cardiac, and smooth muscle. Our focus starting off is going to be on skeletal muscle. That is going to be our major focus. Once we get done with the muscular system, we're then going to move into the cardiovascular system, and we'll talk about the heart and how the heart contracts in order to move blood. Well, and then we'll talk more about cardiac muscle. And then we'll talk about blood vessels that deliver the blood pumped from the heart. Well, blood vessels are lined by smooth muscle. So we'll talk about cardiac and smooth muscle as the term progresses. But we're going to start off with skeletal muscle. So we got pictures of all 3 kinds here. This is a little histological picture of a skeletal muscle fiber. There's another one here and another one up here. Over here, this is cardiac muscle tissue, and here's smooth muscle tissue down here. Now, there are some shared characteristics between the different muscle types. All right, skeletal and cardiac muscle, they're made of 2 primary proteins called actin and myosin. And the way that actin and myosin are arranged in the muscle cells is a very regular arrangement. And it creates these alternating dark, light, dark, light, dark, light bands when you shine a light through it and look at it on a microscope. Well, that alternating dark light bands, it gives the muscle fiber, the muscle cell, a striped appearance, right? So all of these stripes here, they're called striations. So this muscle is striated or striped. Now, in muscle tissue, this is one really long cylindrical shaped muscle fiber, and they all kind of run roughly parallel to one another. And I did mention most of the time these muscles attach to bones at either end. However, there are some exceptions, and we'll run through those exceptions, the muscles that attach to the skin or underlying connective tissue called fascia. Now, if you look really closely at the cardiac muscle, you can see that each one also does this a striated appearance, right? It's the actin and myosin is organized in the same way in these 2 different muscle types. A couple of differences are, one, we have voluntary control over our skeletal muscle fibers. Most people do not have voluntary control over cardiac muscle. Most people cannot voluntarily regulate their heart rate. All right. Okay. Yeah. Sure. There are. It would be remiss if I did not mention that there are established studies, peer-reviewed journal articles that show some people can do this, right? But not most people can't. And we're gonna cater to the 70% of the human population. Most people can't do that. All right. So it's involuntary. And when we get into the heart, we'll talk about how it automatically produces a rhythm, right? In 231, we talked about the nervous system, right? And how cells can depolarize and get excited, or they can hyperpolarize, right? And get less excited. These specialized cells in the heart, they are autorhythmic. They just automatically depolarize, and then they generate muscle contractions, and then they just start to depolarize again. And they're just called pacemaker cells. They set the rhythm of the heartbeat. Right? So skeletal muscles are not autorhythmic. They need to be activated. They need to be told what to do. A skeletal muscle fiber will never, ever, 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 ever do anything unless it is told what to do by the nervous system, by a neuron that communicates with it at what's called a neuromuscular junction. Cardiac muscle is different, right? And whereas skeletal muscle fibers are cylindrical and roughly parallel to each other, cardiac muscle cells are basically stacked up end to end to end to end to end. It's called in series, how they're organized. And this dark line right here, and this one right here, and here, and here, and here, all of those dark lines, they represent junction points between adjacent cardiac muscle cells. And those junction points are called intercalated discs. And they consist of two different types of cellular junctions called desmosomes and gap junctions. But like I said, more on the heart, more on cardiac muscle when we get to the heart. And also, like I said, when we get to blood vessels, same deal. We'll spend more time on smooth muscle. But it's not striated. It's also involuntary. Most people don't have voluntary control over how fast food moves through our digestive tract. Most people don't have voluntary control over their blood pressure, right? By controlling the diameter of a blood vessel. So, like I said, more on that to come. For now, know the three types, know the similarities and the differences between the three types, whether they're voluntary or involuntary, striated or not striated, and an example of where you could find cardiac muscle, pretty easy, and where you could find smooth muscle in the body. All right, now like I said, the focus here is going to be on skeletal muscle. All right, and this is an example of a little cartoon representation of a skeletal muscle and how it is attached to a bone. All right, now the way I was taught how a skeletal muscle is organized, it was kind of like it's like a bundle within a bundle within a bundle within a larger bundle. All right, so let's start all the way down here at the individual muscle fiber. All right, each individual fiber, this thing sticking out right here with the circle around it, that's one muscle fiber that's sticking out. All right, and that represents a muscle cell, a skeletal muscle cell. All right, basics of A&P. Cells are separate from other cells. They're surrounded by a plasma membrane, right? That's what holds all the organelles in and all the cytosol, the fluid. Well, a muscle fiber cell is surrounded by a membrane, okay? And the name of that membrane, shown here in blue, is actually on the next screen. It's called the sarcolemma, all right? But we're going to get to that in just a second. So this whole muscle fiber sticking out is actually made of even smaller components. This part sticking out, and there are these right here. And we'll, like I said, we'll get to it. But this one muscle fiber, right? Here's another muscle fiber right here. There's another one, and another one, and another one, and yep. All of these little dark areas right here are muscle fibers or muscle cells. And this muscle fiber is separated from this muscle fiber, which is separated from this muscle fiber by connective tissue, right? And that connective tissue is called the endomysium. So the endomysium separates individual muscle fibers from each other. Within all of these muscle fibers, they're all part of a larger structure, this whole thing sticking out right here, which is called a fascicle. Right? So this one fascicle that's sticking out is made up of multiple muscle fibers. All right? And here's one fascicle sticking out. Here's another fascicle right here. And here's another fascicle with more muscle fibers in it. Here's another fascicle with more muscle fibers in it. Yeah. And yeah. And yeah. Again and again and again. Right? So you get the idea. Well, just like a muscle fiber is kept separate from other muscle fibers within a fascicle by way of endomysium, a fascicle is kept separate from other adjacent fascicles within a muscle by way of connective tissue. But this connective tissue is called the perimysium. So the perimysium surrounds this fascicle, keeps it separate from this one. All right. Now, all of these fascicles they're also surrounded by and like a little sheath of connective tissue that wraps around all of these fascicles. That sheath is more connective tissue, and it's called the epimysium. All right, and then seriously, just a lot of organization here. And even then, surrounding that, there's another thin sheet of connective tissue called the fascia, which actually merges with the dense regular connective tissue, all of the collagen fibers that make up a tendon, that then attach this muscle to the bone. And that tendon and the deep fascia, it's actually continuous with the membrane that surrounds and wraps around a bone. It helps anchor it strongly to that bone. Right, so that, and just a reminder, the layer of connective tissue that surrounds a bone is called the periosteum. Peri means around, osteum refers to bone. All right, so one more time, right? Let's walk our way through this. A whole muscle is surrounded by deep fascia. It's attached to bone by way of a tendon, surrounded by deep fascia. If you get deep to that, the whole muscle is again surrounded by an epimysium, which holds all these fascicles together. Each fascicle is separated from another fascicle by perimysium, and within each fascicle there are multiple muscle fibers, each kept separate from others by way of endomysium. And then each individual muscle fiber has a plasma membrane, which is then called a sarcolemma. Which brings us to this, the structure of a muscle fiber. Right, so I go back again. This thing sticking out here, one muscle fiber, that's this. Right, a magnified view of that muscle fiber from the previous slide. So this blue thing right here that's peeled away, it's labeled sarcolemma. That's the plasma membrane. If you open up that sarcolemma and look inside a muscle fiber, it's made of even smaller units called myofibrils. And a muscle fiber is sometimes called a myofiber. This is a myofibril within a muscle fiber. This cartoon shows what? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 different myofibrils that make up this myofiber. Please don't remember the numbers of that because it's ridiculous. Right? You'll never be held accountable for how many myofibrils make up a muscle fiber. It's crazy. Okay. All right, but take a look at this sarcolemma. All right. It is punctuated. Look, these little openings right here. There's a little dot right there. There's one here and here and here and here, and there's one pointing it to it here. There are holes in that sarcolemma, openings that allow ions in the extracellular fluid that exists outside the muscle fiber to make their way into a muscle fiber and possibly lead to a contraction. Okay, so these openings in the sarcolemma, they actually empty into a little tube, which is shown here it's the purple one. All right, that is called a T-tubule or a transverse tubule, which kind of makes sense how it's organized with respect to the myofibrils. It's kind of perpendicular to the orientation of a myofibril, which is transverse. So ions pass through these openings in the sarcolemma, and they then enter this transverse tubule. There's one of them there. Here's one of them shown here. Here's one shown here and one shown here. Right? Different examples of a T-tubule. All right. I'm basically what I'm going to do here today is I'm going to throw an amazing amount of anatomy at you regarding a muscle fiber, and then once I've thrown all of this anatomy at you, I'm going to put it all together into the physiology and what all of these anatomical components do and how they do it. Okay, but we got to get through the anatomy first. All right, just lateral to each T tubule or transverse tubule are specialized tubes, ducts, if you will, that store a lot of calcium. Right? They're called cisternae. A cisternae, it's like a cistern is kind of like a cup, right? So on either side of the T tube, you will have what's called a terminal cisterna. There's this blue one right here, and there's one on this side of the T tubule as well. If you take one terminal cisterna and make it plural, then it's terminal cistern E. This means that there's two. Okay, it's just Latin. Plural in Latin, you add an E to the end of the word. So these three structures, the one T tubule and the two terminal cisternae that flank it, collectively are known as what's called the triad. All right. And then this terminal cisterna on this side is connected to the terminal cisterna that's part of this triad over here. And it's connected by way of this labyrinth of tubes called the sarcoplasmic reticulum. Right? Most animal cells have what's called an endoplasmic reticulum that's involved in protein and lipid synthesis, but it's a specialized plasmic reticulum in muscle called sarcoplasmic reticulum, and it stores a lot of calcium ions, which are, you're going to find out, critical for muscle contraction. All right, let's see. So I got the myofibrils, we did the sarcoplasm, that's pretty self-explanatory. It's like the same as cytoplasm, just specific to a muscle cell. The cytoplasm is all the fluid as well as the organelles within a cell. So this is just everything that exists in between the plasma membrane. That's sarcoplasm. All right. Now, we got lots of mitochondria. Hopefully you remember what mitochondria are. They're known as the powerhouse of a cell. The more mitochondria you have, the more ATP you can produce with oxygen present. So a lot of mitochondria, a lot of ATP, a lot of energy. Skeletal muscle tissue demands a lot of energy. So there are lots of mitochondria here. All right. Each one of these myofibrils that collectively make up a muscle fiber is in and of itself made up of even smaller subunits that are stacked up end to end to end. And each one of those subunits is called a sarcomere. Okay? Now, there are these terms here, you're going to have to know them. I'm going to go through them, but Z-disc, A-band, I-band, here are a couple of terms. We're going to add some more onto that, right? College, it's hard. This Z-disc, it represents the end of a sarcomere and the beginning of another one. So if you go from this Z-disc all the way over to this Z-disc, that area in between is one sarcomere. Where this Z-disc is to the next one, that would be the next sarcomere. So each myofibril is a sarcomere stacked end to end to end to end. All right, now let's take it a little bit further. This is showing this right here, one sarcomere. All right, so we're taking this image, we're taking one of these myofibrils and bringing it out over here. This is it, the myofibril. And so here's a Z-disc. Z-disc, Z-line, I don't care. Either way, call it a disc, call it a line, means nothing to me. All right, so from this Z-disc to this Z-disc, that, my friend, is one sarcomere. All right, and the next sarcomere would begin right where this one ends and go to the next Z-disc. And then the next, and then the next, and the next. Okay, what each of these sarcomeres is made of is primarily two major proteins called actin and myosin. Each of them is made up of smaller, protein units, and we'll get to those, tropin and tropomyosin. But actin and myosin are the major players here involved in contraction. All right, now, actin in this cartoon, and we take this sarcomere and kind of blow it up and enlarge it, that's what we're looking at down here. Here's the Z disc right there at one end. There's the Z disc at the other end. This whole area equals sarcomere. All right. Actin is shown here in yellow. We'll spend some more time on it. It's coming, but actin's shown here in yellow, and it's thin. It's thinner than this, which is myosin. So actin is known as the thin filament. There's some actin here. There's here. There's here. Here's some actin associated with the next sarcomere. Same thing over here. And there's actin here, here, and here. And this, please understand, this is a two-dimensional representation of a three-dimensional structure. So it's just not. You can say that it's complex and hard, and I'll agree with you, but it's so much more complex than what's shown here. Okay. And you can see that the actin strands, they're actually anchored to the ends of a sarcomere. They're anchored to the Z discs at the end. They don't cover the entire, they don't span the entire sarcomere, right? The myosin, same way. The myosin, thick filament, it does not span the entire sarcomere. It's found in the middle part of the sarcomere. At the ends of this end and this end, there's no myosin there. You've only got actin as well as a spring-like protein called titin. That's why it gives this coiled look right here, like it acts like a spring, right? Which gives muscle, skeletal muscle, one of its inherent properties, which is elastic, all right, as well as so it can be stretched beyond its resting length, and it has recoil. Right, if you stretch it beyond its resting length and let it go, it'll snap back kind of like a rubber band. All right, that's the role of titin. It helps anchor myosin to the ends of the Z disc, or to the ends of the sarcomere, at the Z discs. All right, so there's actin, there's myosin, there's titin. One that's not shown, dystrophin. Okay, there's not going to be a picture of dystrophin anywhere in the lecture slides. But what dystrophin is, it is another muscle protein that actually helps attach the myofibrils to the plasma membrane within a muscle cell. It kind of serves as like an anchoring point for the myofibrils. Sometimes people don't produce adequate dystrophin or don't have it in the amounts that they need it, or for some reason it's attacked and it's destroyed and it degrades. And people can present with conditions like muscular dystrophy. It's a lack of dystrophin. If your myofibrils aren't anchored to the plasma membrane, they can't produce the same amount of force as if they were. So you get weakness. It's a classic sign of muscular dystrophy. And if you can't contract the muscles, they can't exercise to the same amount, the same level. They tend to atrophy, and you get muscle wasting as a result. And if you just maybe not seem like the end of the world, but if it affects your diaphragm, which is breathing, and you can't generate adequate force to inflate your lungs, well, that's bad. Okay, so come back here. I mentioned these three terms, Z-disc, A-band, I-band. We got the Z-disc covered already, right? It's the end of a sarcomere. Couple other terms for you to know. One, the M-line. M-line right here. It's a protein. It's actually a protein called myomycin, shown here, that quite literally bisects the sarcomere. It runs right through the middle of the sarcomere and serves as kind of another anchoring point for these myosin myosin proteins. All right, now if you're looking at a sarcomere up here, right, and say you've got this area over here where it's light and then dark and then kind of light again and then dark and then light. Right? That's the organization of actin and myosin. Where actin and myosin overlap, if you shine a light through it, that overlap is going to make it appear darker. Right? At this area here, you don't have the overlap. It's only pretty much just actin. More light shines through it. It appears lighter. Right? So, these terms here, the A band, I band. The A band is basically starts right here where myosin begins and ends at this end within a sarcomere where myosin ends again. So this whole area, right, from here to here, that's where you'd find all the myosin. That's the A band, right? So from here, A band shown here, from there, all the way over to there, that is the A band. Where myosin ends, now you have this light region, then a Z disc, and then there's going to be another lighter region over here until you run into more myosin from the adjacent sarcomere. So this area here that's lighter is called the I band. Right, so basically, if I come back here, this area here, I band. Dark, A band. Light, I band. A band, I band, so on and so forth. So we got M line in the middle, Z disc at either end. The I band, which is the light area here, bisected by the Z disc, and the A band, which is the darker area, bisected by the M line. And the only one left here is this center region in the middle of the sarcomere where there are no myosin heads. There's no overlap between actin and myosin. That area is called the H zone. I did tell you, I'm gonna throw a lot of anatomy at you first before I put it all together. All right, so even more in depth here with actin and myosin. Start with myosin first, known as the thick filament. Right, each one of these. Right, this is a two-dimensional view of a sarcomere. Right, each sarcomere can have 200 to 500 of these myosins, these thick filaments in there, all surrounded on all sides by actin strands, actin filaments. Okay? But basically, what myosin it is, it's a polypeptide. We get these, these two proteins that wrap around each other, and they have these two little they look like golf clubs. That's how it was shown to me or explained to me. They look like golf club heads that just stick out at either end. And all of these myosin heads are directed outward in like this kind of spiral arrangement where all the heads stick out. All right, now, where all these heads stick out over here, that's analogous to this over here. There's an overlap. That's the area that overlaps with actin, which is going to be extremely important coming up. All right, and this here, there'd be the M line running through that, made of myomesin protein. And then this whole area right here, just to recap, this would be called the H zone, the center region. No myosin heads there. Now we get to actin. Actin's actually sometimes referred to as the actin complex because you got three different proteins that make up, that contribute to this. Actin, troponin, and tropomyosin. All right, now the actin strand itself, actin is actually made of these little globular subunits called globular actins or G-actins. So that's one, boom, 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 boom. And all these G-actins are kind of arranged in this helical array where they kind of twist around each other. Each one of those individual G-actins has a binding site on it for a myosin head. It can allow a myosin head to attach to it. Okay? Now, normally, if a muscle is at rest and it's not producing force, it's not contracting, all of those myosin binding sites, they're all covered by this thin thread-like protein right here called tropomyosin. Right? They're covered. Myosin can't bind to them if a muscle at rest because tropomyosin's in the way. All right. Now even further, attached to each one of those tropomyosin molecules is another protein called troponin. All right. And just like a G-actin has a binding site for a myosin head, troponin has a binding site for calcium. It can bind calcium ions. Okay, here we go. We're going to start to put the whole thing together now. Here we go. All right, like I mentioned, a skeletal muscle fiber never does anything unless it's told to by the nervous system. All right, so where nervous tissue communicates with muscle tissue, that's called the neuromuscular junction. All right, so here's the end of an axon of a motor neuron, and it contains little sac-like structures called vesicles that contain molecules of neurotransmitter. Okay, and the only neurotransmitter that is released at a neuromuscular junction that can lead to muscle contraction is called acetylcholine. So you'll sometimes see it as ACH. ACH, acetylcholine. All right, so an action potential, right, travels down here. It causes the release by way of exocytosis. It will cause the release of acetylcholine. Acetylcholine released into the synapse will then cross the synapse and bind to specific receptors. Acetylcholine receptors on the muscle membrane. Okay? On the muscle cell itself. All right? Now, that binding of acetylcholine is going to open up voltage-gated sodium channels that are also in the muscle membrane, shown at point number 2 right here. And sodium is going to follow its concentration gradient from the extracellular fluid into the muscle cell. All right, so sodium is going to flow in and it's going to depolarize that muscle cell. That wave of depolarization is going to spread along the sarcolemma. And as it spreads along the sarcolemma, it's going to invade those openings that lead into T-tubules, right? The holes in the sarcolemma. Right, so as the action potential passes down these T-tubules, they are lined with voltage-gated calcium channels, and that's going to cause. And just on either side, remember you have the terminal cisternae right here, shown in green, the calcium storage facility of the sarcoplasmic reticulum. Right, so all this, if you open up voltage-gated calcium channels, and there's a lot of calcium stored in here, it's going to follow its concentration gradient out of the sarcoplasmic reticulum into the muscle cell. Right? So all of these steps, the acetylcholine, the sodium, the depolarization, they all are going to function to cause the release of calcium ions inside the muscle cell. And in this cartoon image, the green here represents myosin, and the purple lines represent actin strands, and the actin is attached here to a Z disc. Great. And so there's that. So what happens to that calcium once it's in? Well, if you recall, troponin, right? Troponin has a binding site for calcium ions. Right, so here's the G-actins. There's the tropomyosin strand that has troponin attached to it. So calcium is going to bind to that troponin. And when it binds to troponin, it's going to cause troponin to change its shape. Troponin will actually shift and move to the side. And because it's attached to tropomyosin, it will drag the tropomyosin strand over with it. And when tropomyosin gets dragged out of the way, it exposes binding sites for myosin heads that are normally covered. And sitting right there, there's myosin waiting for a chance. Like, open the door, let me bind to this binding site. So calcium allows for myosin to bind to actin. And it's the binding of actin, that physical connection between actin and myosin, that's called cross-bridge formation. It's a physical connection between actin and myosin. Okay, lot going on this slide. All right, but let's pick it up. Let's pick it up at point number 2. Okay, this is where ATP comes into play, and ATP is over here. Okay, but let's just pick it up right. Okay, fine. We'll start at number 1. All right. actin here is shown in purple. There's the thin filament. Here's myosin, the thick filament. In green, there's a myosin head. All right. Now, when myosin is waiting to attach to actin, it's in what's called an energized state. Means that the myosin head is ready. It's energized. And because of that, it took an ATP molecule to get kind of broken down, I guess, hydrolyzed, if you will. So ATP is kind of hydrolyzed into adenosine diphosphate, and one of those inorganic phosphate groups is broken off from it. Okay? And that's what creates this energized state. When calcium floods in, binds to troponin, exposes the binding sites, you're going to get cross-bridge formation. Myosin will attach to the actin strand. And in doing so, it will reduce its affinity to hold that inorganic phosphate. It will release that inorganic phosphate through the action of binding. This is kind of like a one causes the next, causes the next, causes the next kind of thing. All right. So once that inorganic phosphate is released, that's then going to cause the myosin head to pivot, to change its shape. Right? The myosin head is actually going to pivot inward towards the middle of the sarcomere, towards the M line. And because it's attached to actin, it's going to drag actin inward as well. This is called the power stroke. It's the relative sliding of these two filaments past each other. Okay, and that again, that pivoting action of the myosin head inward towards the middle of the sarcomere, that's going to then cause that adenosine diphosphate molecule to be released. And when it does, that will then, that will allow for an ATP molecule to bind to that spot. And once ATP binds, ATP breaks the cross bridge. It literally separates actin from myosin. And then that ATP molecule is hydrolyzed. One of those phosphate bonds is broken. That puts it back in this energized state. And if there's still binding sites available, myosin will attach again and go through this entire sequence called excitation, contraction, coupling. And it will continue as long as calcium is binding to troponin, causing myosin binding sites to be exposed, as long as ATP is available to continue breaking cross bridges and allowing this to continue. All right, so again, like I said, I am not going to read all of this to you, okay? You're in college, man. You know how to read. But you can pick it up with step 1. This walks you through everything that was just gone over. From the nerve impulse arriving to acetylcholine release, its binding, what happens at the T-tubule, what happens to actin and myosin, the power strokes, and all of this. All right, now, this is what's behind rigor mortis. And rigor mortis is the stiffening of the body that occurs after death, post-mortem. I mean, if you think about it, it doesn't happen right away. And it's not immediately. It's soon after death that the body stiffens and gets to rigor mortis. Well, why does the body stiffen if you die? Well, I mean, what's that about? It's lack of ATP production. Right? If you can't deliver it, all right, your heart stops beating. You're no longer delivering blood and oxygen to your muscles. Okay, pretty obvious. No oxygen, you're going to have a drastic reduction in ATP production within a muscle. And without ATP, well, you can't break cross bridges, right? So you get this constant contracted state in muscle due to the lack of ATP and the inability to break actin and myosin cross bridges. Now, eventually, sure, those proteins will degrade and decompose and liquefy and all the good stuff that goes along with post-mortem, with dying. So there's that. Now this, I haven't yet, but I will. I will post this animation to Canvas so you have access to it. This is just something kind of cool. If you look at it, it's really easy to let your eyes glaze over. But take a second, all right? Let me walk you through this right here. So the yellow, each of those yellow, they represent globular actins, so G-actins, all right? The blue strand, that represents tropomyosin. The fuchsia / purple snowman looking structure, that's going to be troponin. And the yellow dot swooping in from the top right, that's going to represent a calcium ion. Right? So follow that calcium ion. It's going to come in. It's going to bind to troponin. When it binds to troponin, troponin moves. Right? When troponin moves, it drags tropomyosin with it. The blue strand moves. It moves. When tropomyosin moves, boom, myosin can attach. All right, so what's happening at the myosin head? That full green circle represents ATP. When it binds, you'll see myosin detaching. Cross bridge is broken, right? Binds, boom, breaks. Boom, 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 boom. All right, and then I did not cover the cone-shaped gray structure. That is something called ATPase. It is an enzyme that actually breaks that phosphate bond on ATP and makes it ADP and the inorganic phosphate. And you can see that inorganic phosphate kind of frisbeeing away, boom, 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 after it's been broken and after the cross bridge has been formed. Right, so if you can pick out different pieces of this animation, it can be helpful. You are going to have to open this in slideshow mode within PowerPoint in order for the animation to play. You can't just open the PowerPoint and look at the slide. That won't work. You got to put it in slideshow mode. All right. Muscles can produce varying amounts of force, right? A given skeletal muscle, it can produce, there's a maximum amount of force that it can produce, right? The maximum, and that amount of force that it can produce is a direct reflection of how many cross bridges are being formed within that muscle. All right, so in this case, on the y-axis, you've got percent of total force production. So 100% of force production, and it matches up with the resting length, the optimal length of a given muscle. All right, and at this orientation, the pink represents myosin, the blue dots represent actin. There's a Z disc. There's a Z disc. All right, at this muscle length, you've got maximum overlap between actin and myosin, and all available cross bridges could potentially be formed, which is the maximum amount of force you could produce. Now, say you, if it's the elbow, say you flex your elbow, right? So now you've shortened the muscle. That's equivalent to this. You've moved the Z lines closer together. So the actin that's attached to those Z lines is going to overlap the H zone, the center of that sarcomere. And in the H zone, there are no myosin heads. So all those actin, or the binding sites on actin for myosin heads are wasted because there's no myosin there, no myosin head to bind to it. You have fewer cross bridges. Fewer cross bridges, lower force production. The same thing. You mean if you say you lengthen, you extend your elbow and straighten your arm all the way out. Well, basically what you're doing is you're taking those Z discs and you're pulling them further apart. So now you've got these areas at either end that are part of the I band, right, that aren't overlapping with myosin. These are wasted cross bridge sites. So once again, you've reduced cross bridges, you have reduced force production capacity. That's the force-length relationship. Direct reflection of how many cross bridges you can form. All right, now I'm talking about muscle contractions. Right? Not all muscle contractions are exactly the same. There are 3 different types of these muscle contractions, and they're listed here: concentric, isometric, and eccentric contractions. All right, now, and they're kind of listed here as well. if you were in anatomical position, all right, anatomical position, arms down at your sides, and you're holding on to a weight in your right hand, right, and you produce sufficient force in your biceps brachii muscle. All right, so this is going to be the agonist muscle, the one actually producing the action, the biceps brachii right here. It will flex the elbow and create this angle. And so it's producing force, and the muscle is shortening while it's producing that force. That is a concentric muscle contraction. Now, say you're holding on, and if you say you flexed your elbow and you got to this position and you held it, no movement, you have to still produce force to resist gravity, wanting to extend your elbow, wanting to straighten your arm and pull that weight to the ground. You have to produce the exact amount of force to counter that gravitational force. Right? So if you do that, it's force production, but the muscle isn't changing length. Iso means the same, metric means length. So an isometric contraction, force production, no change in muscle length. All right, now this is the one that trips people up sometimes, all right? Eccentric contractions. Now, say you're holding on, say you're in this position right here, all right? You're holding on, you're holding their elbow at a flexed position. If you stopped producing any and all force, gravity would just, straight extend your elbow and straighten your arm, right? But what if you wanted to extend your elbow and straighten it slower than what gravity would do on its own? Right? That's an eccentric contraction. You have to take the amount of force you're producing with an isometric contraction and slowly turn that force production down. And as you do, you can gradually allow the gravitational force to exceed the muscular force, and you can slowly extend the elbow. The muscle is still controlling it, but the muscle is lengthening while it's producing that force. That is an eccentric contraction. I'm not a gym rat, okay? If you are, rock on for you. Great workout, super duper. All right? But, I mean, it is known that if you are attempting to, if your goal is to build muscle mass just to get, like, beefy, you want to do an exercise program that is loaded with these eccentric contractions. They actually result in greater muscle hypertrophy or increase in size of the muscle. Not necessarily strength, but you'll look bigger. So, I don't know. We all have goals. I don't know. If that's your goal, I'm not here to tell you anything good or bad about it. Okay. Now, just like there are different types of muscle contractions, there are different types of muscle fibers, even within skeletal muscle. Okay, you've got, they're largely named or classified according to their fuel source, the primary fuel they use to generate muscle contractions. So if it uses oxygen as a primary fuel source, it's known as an oxidative fiber. Okay. This should not be brand new. The material, okay, should be new. But it should not come as a surprise anyway, that in anatomy and physiology, there are often multiple terms for the exact same thing. And here's a prime example of this, okay? A muscle that uses oxygen as a primary source of ATP production, it's known as a type 1 muscle fiber. It's also known as an oxidative muscle fiber. It's also known as a slow twitch muscle fiber. I don't like it either, but it doesn't change the fact that that's what it is. If you have a muscle fiber that primarily uses glucose or glycogen as a source of ATP production, that's known as a type 2 muscle fiber, or a glycolytic muscle fiber, or a fast twitch muscle fiber. And you can actually find out, you can go through a process called fiber typing, where you can actually go in. I've had this done to me, and I will tell you that it is remarkably uncomfortable to have this procedure done, but that's life. It involves getting a sample. You insert this large syringe into the muscle, apply local anesthetic, of course. Then you pull the plunger back, it opens this trap door, and you push the plunger down, it snaps that trap door shut, and it snips off a little bit of your muscle fiber, and you pull that out, slice that fiber, that muscle tissue up, and stain it, and look at it under a microscope. All right? And what you see is you see either dark or light stained cells. Each one of these is a cross-section of an individual muscle fiber. Right? Those that stain dark, that means there's a lot of blood vessels that supply that muscle fiber. What's carried in blood? Oxygen. So these dark muscle fibers are oxidative muscle fibers, type 1. Those that are white do not have the same capillary density. They don't rely on oxygen to the same degree. They rely more so on glucose and stored form of glucose, which is glycogen, and they show up as white muscle fibers. This is exactly, if you don't eat meat, okay, don't eat meat, I don't care. But if you do eat meat, like chicken or turkey, things like that, some people like dark meat, some people like white meat. Some people my family at least gets grossed out when I bring this to their attention. But if you like the dark meat on chicken or turkey, you like to eat type 1 muscle fibers. If you like the white meat on chicken and turkey, you like to eat type 2 glycolytic muscle fibers. Kind of morbid to think about, but doesn't make it untrue. Okay, now here's a little table, a little rundown of just some characteristics with which you should be familiar of these different fiber types. The type 2s can actually be even further broken down into type 2As and Bs. Type 2B, this is almost exclusively glycolytic capacity or glycogen use. Very, very low oxidative capacity. All right, so type 2Bs, the muscle fibers are really large relative to a type 1 oxidative fiber. Small, right? Small versus large. A small muscle fiber has less protein in it than a large muscle fiber. So fewer proteins, fewer cross bridges, low force production relative to a type 2 fiber that are bigger. Right? The contraction speed is slow. Slow versus fast, this is all relative. All right? The contraction speed of a type 1 muscle fiber is about 100 milliseconds, a tenth of a second. The contraction speed of a type 2 muscle fiber, it's about half that, about 50 milliseconds. Right, so twice as fast as a slow twitch fiber. The glycolytic capacity, I like to think that makes sense because glycolytic fibers have a high glycolytic capacity, and that same logic should hold true down here for oxidative fibers. They're going to have a high oxidative capacity. Where do you get that oxygen? Blood, capillaries. All right, and because now the fatigue resistance, this is again something that sometimes trips people up. How long can that muscle fiber continue to contract before it just fails, it fatigues? Type 2s, you get a lot of force and you get it right away, but it doesn't last very long. It's got a very low resistance to fatigue, which means it fatigues quickly. Type 1s, they're pretty small, they take longer to contract, they don't produce as much force, but you can keep using them for a much longer period of time before they fatigue. This is what gets back to this. You see a lot of the dark fibers, the type 1s, long distance running, right? High resistance to fatigue. A lot of type 2 white glycolytic fibers, more suited for sprinting. I need the power, I want it right now, and I want it, all of it now. I don't want to have to spread it over like a mile. I just want to do it for 100 meters. All right. And then I mentioned that skeletal muscle fibers never, ever do anything unless they're told to. Well, that's true. Here's a motor neuron, and a motor neuron that communicates with a skeletal muscle cell, it's called an alpha motor neuron. It's spelled out there as alpha, just shorthand here, alpha motor neuron. This one motor neuron can innervate or control numerous individual muscle fibers. All right, so you take that one motor neuron and all the muscle fibers that are controlled by it, say in this case, these 5, which is ridiculous, this right here represents what's called a motor unit. A motor unit is a motor neuron, an alpha motor neuron, and all the muscle fibers it innervates. That is the definition of a motor unit. Now, some motor units have 10 muscle fibers. Some have 3,000. Some have like 10,000 fibers. Like the muscles that move our eyes, that are attached to our eyes, one motor neuron can control 10 muscle fibers, which gives us the smaller the ratio, the more fine control, the more graded force production you can produce, which is awesome for eye movements or for really small muscles, right? But if it's say your gastrocnemius, or your calf muscle, motor units in the calf, or what's called the sural region. I won't say calf again because it just hurts me to say that. The sural region, I mean, they can that one motor neuron can innervate up to 3 to 5 to possibly 10,000 muscle fibers. And every time one action potential is sent along this one alpha motor neuron, all of the muscle fibers will contract. They're all going to be type 1s or type 2As or type 2Bs within a motor unit. Right? But say if this had 10,000 muscle fibers and all of them contract at once, well, that does not give you fine graded force control or force production. That gives you gross force production. And then this motor unit, combined with all the other motor units that collectively innervate and control all of the muscle fibers within a given muscle, that is called a motor neuron pool. And when we want to contract a muscle, we go through an orderly recruitment of these motor units. We recruit the small ones. It's like an energy-saving mechanism. Why activate the big motor units if you don't need to? You're going to waste that energy, right? So you want maximum production with minimal energy expenditure. That's the goal. So when we want to move, we'll recruit the really small motor units first that control type 1 muscle fibers. If that's insufficient to meet the demands of the task that we're trying to perform, then we'll recruit the next largest size motor unit. We'll add on more muscle fibers to the task. That will increase force production. That might be sufficient. If it's not, well, then we activate the next largest type or size of motor unit that controls the type 2B's muscle fibers. Right? So it's an orderly recruitment of motor units. All of this, the motor unit recruitment, it falls under the umbrella term of what's called the Henneman size principle. I'm going to make sure it's spelled correctly. Yeah, it is. Henneman's size principle. It's spelled correctly in the closed captioning. So this is the Henneman size principle, the recruitment of motor units from small to large, and the de-recruitment of motor units at the end of exercise in the order of large to small. You turn off the big ones first, and then the medium ones, and then you turn off the smallest ones at the end. All right. That, I believe, I am going to save for the next one. All righty. We figure this out here. I will figure this out. Maybe I will. Maybe I won't. I think I did it.